Hello everyone and welcome to another recorded lecture of BIO101 online. Today we will be discussing chapter 13, which is on gene expression. So I want to talk about the genetic basis of pigmentation as just a brief introduction to gene expression. We discussed in the past two chapters that all life on Earth contains the same four bases of DNA. We have adenine, guanine, cytosine, and thymine. And together, these four bases are molecules that make up the genetic code that encodes for proteins that make up every cell in our body. Like different combinations of the A's, T's, C's, and G's make up every gene in our genome, right? And we have about 20 to 30,000 genes that code for proteins. One of those genes is called MC1R. It's actually a receptor. And it contributes to skin color, hair color, and eye color in humans. In every single cell of the human body, if you opened up the nucleus, you would see the MC1R gene. Right? It's found in the nucleus of every somatic cell of the body. Even if it's a neuron, right, MC1R is still there. Only some cells, called melanocytes, actually require that protein though. Right? You don't really need a hair color gene in a neuron or in a muscle cell, right? Only certain types of cells called melanocytes rely on melanin for pigmentation. And humans, right, have a lot of variations in that MC1R gene that come from their ancestry. So depending on the exact sequence of your MC1R gene, you might be a redhead or you might be a brunette. So not only does not every cell in your body express it, but individuals express different versions of the same gene. So we're going to talk in chapter 13 a lot about the differences in DNA-based sequences and how they alter gene expression. Right, so in this case, we're talking about pigmentation. The sequence of this gene can be mutated, which can lead to different phenotypes. And then within an organism, right, within the human organism, every single cell has every gene, but not every single cell will express that gene. All right? So everyone has melanin genes, but each individual's gene expression is determined from information in their genome, and also their environment contributes as well. All right? So just to reintroduce this, we said the genome of an organism has all of the genes required for it to function. And there are about 20 to 30,000 protein coding genes in the human genome. So are all the genes in a genome expressed in every cell? Right? We said no, right? Not every gene gets transcribed and translated into a functional product. Right? Only certain genes are expressed in specific cell types at a given time. So gene expression must be very highly regulated in all organisms. Right? If every cell of an organism has every single gene, there must be a lot of coordination and control about which genes get turned on and turned off in each cell type. Right? Because proteins and enzymes should only be produced as needed. Right? Why would a neuron waste its time making a hair-colored protein? I want to just quickly talk about prokaryotic gene regulation. I'm not going to focus too much about it on exams. I just want you to be a little familiar with how prokaryotes regulate gene expression. So the example I want to talk about is called the trip operon. And in bacteria, the trip operon is a group of genes that regulate enzymes that make the amino acid tryptophan. So as you recall, there are 20 amino acids that build proteins. One of those 20 amino acids is called tryptophan. And bacteria can either make their own tryptophan, or they can use tryptophan from an outside source. So as you could imagine, every bacterial cell has genes that can make tryptophan if needed. Right? So when tryptophan levels are low, genes that make tryptophan will be turned on. Right? That should make sense, right? The genes that can make this amino acid will be turned on if there's low tryptophan levels. right? The bacteria will sense the low levels and say, okay, I need to make more tryptophan. But what will happen if there's already enough tryptophan around? Should the bacteria still be making more tryptophan if the tryp levels are already that high? So what happens is that if tryptophan levels are high, there's a shift in gene expression. Tryptophan itself, the amino acid, combines with a special protein called a repressor protein. So the actual tryptophan, when there's enough of it, there's so much of it, it combines with a protein called a repressor protein. And the repressor protein is only functional when tryptophan is present. And what happens is that the repressor will block the synthesis of enzymes that can make more tryptophan. So this is a good feedback loop. This is like a negative feedback, right? If there's enough tryptophan, it will bind with the repressor protein to stop the production of more tryptophan. When tryptophan levels are low again, the repressor cannot attach to where it was blocking transcription because there's no tryptophan, right? Remember that the repressor requires tryptophan in order for it to stop transcription. So therefore, if there's no repressor, RNA polymerase is free to bind the promoter and transcription of those enzymes can be initiated. So this is a quick schematic just showing how there's a separate gene outside of the tryptophan producing genes that makes a repressor. So let's just look over here. Let's look at the promoter first. Here's the promoter. This is where RNA polymerase will start transcription. Then there's a little sequence called the operator. And then here is one group of genes. In bacteria, they have all the genes lined side to side with no intervening sequences. It's pretty simple. So let's say there are 5 different enzymes required to make tryptophan. So what will normally happen is RNA polymerase, when tryptophan levels are low, will bind to the promoter and will transcribe this unit of genes into mRNA. The mRNA will then find a ribosome, and all these different enzymes will be produced, which can then make tryptophan for the bacteria. However, when there's a lot of tryptophan around, it combines with the repressor, and the code for the repressor is far away from the actual coding sequences for the tryp enzymes. So there's another protein being made far away that can bind to tryptophan when the levels are high, and that will kick off the RNA polymerase. RNA polymerase cannot bind to the promoter when there's an active repressor. So again, this is a very, very, very short example of how bacteria can regulate their gene expression. But the rest of the time, we'll talk about eukaryotic gene expression. So, quick question. Do all somatic cells contain the same DNA? Pause here and see if you can answer that question. Right, the answer is A or C, because it's a tricky question, and that's why I put this as a rapid response. Almost all somatic cells of your body, meaning all cells other than sperm and egg, have the same exact genes. Of course, each cell might have its own mutations, but for the most part, all somatic cells have the same DNA, the same genes, right? They have the same genes. Every one of the 20,000 plus genes of the genome is found in every single cell of your body. So almost all somatic cells, right, almost, have the same exact genes, but only certain genes are expressed into mRNAs and proteins. And that's depending on the type of cell. So for example, all somatic cells in your body contain genes for both alcohol metabolism and neurotransmitters. Those are two very different things, right? One has to do with neurons, one has to do with the liver. But, and right, so even your skin cell has these enzymes, alcohol dehydrogenase and a neurotransmitter gene. But not all cells will express those genes. So for example, in a liver cell, we want to detoxify alcohol. That's the job of the liver. So the alcohol dehydrogenase gene in the nucleus will be transcribed. And that will then be produced into active enzymes that can then break down alcohol. The neurotransmitter gene is still present in the nucleus, but it will not be transcribed by RNA polymerase, and there will not be any neurotransmitters in the liver. Right, in the neuron, again, inside the nucleus of a neuron, we will see both genes. But the alcohol dehydrogenase gene is not required in a neuron, so that's going to be shut off. That gene won't be expressed. Only the neurotransmitter gene will be transcribed into RNA, which can then be translated into neurotransmitters. So expression, right, gene expression means the DNA is being transcribed and translated into a functional protein. And while each cell has a unique package of mRNAs and proteins, all cells have a group of proteins in common, and we call those proteins housekeeping proteins. All cells, whether you're a hair cell, a neuron, a muscle cell, that cell needs housekeeping proteins to survive. These include proteins required to break down biomolecules and build biomolecules. So every cell, whether you're a skin cell, a muscle cell, hair cell, needs proteins and needs to make DNA. Right? So obviously you're going to have all these enzymes required for DNA synthesis. You're going to have ribosomal proteins. All cells need some cytoskeleton. Right? All cells need structure. So every cell is going to have actin and tubulin. So housekeeping proteins is a large group of proteins that every cell expresses in order to survive. They're always turned on. So I want to just do a quick pop quiz. Do you think a heart cell will have heart genes? I want you to tell me yes or no. And you should be answering yes, right? Obviously, a heart cell contains heart genes. Will it express the heart genes into heart proteins? The answer is yes. It will. Will it transcribe skin genes? Or sorry, I should say, does it contain skin genes? The answer is yes. Even a heart cell is going to have skin genes. Is it going to express those genes into having skin proteins? No. A heart cell does not need skin-related proteins. So just because the heart cell has skin genes, it's not going to have the skin proteins. Right? The heart cell will also have neuronal genes, but will not be expressing those neuronal genes into proteins. And of course, a heart cell requires housekeeping proteins in order to survive. So pause here and try to go through the remaining three types of cells and tell me if it will have the genes or the proteins in each of these columns. So here you can see are the answers. All of them have housekeeping proteins regardless of the cell type. All of them have neuronal genes and skin genes and heart genes regardless of the cell type. But not all the cells express the same proteins, right? Express the same genes into proteins. Right? That depends. So now we're going to talk about what determines which genes are expressed and which genes are not expressed. Right? That's the whole point of chapter 13, is what determines whether a gene will be turned on and expressed or turned off and not expressed. So that's the question we're trying to answer. So the answer is not an easy one. So there are several mechanisms that control eukaryotic gene expression. And to make a very long story short, there are certain signals from outside of the cell and inside of the cell that will regulate which genes are expressed in a given cell at a specific time. These signals from the outside might be hormones, right? So from the bloodstream, could be growth factors or other chemical signals, can interact with the cell and tell the cell, turn this gene on or shut that gene off. Also, the inside environment of cells will determine which genes get turned on or off, right? So if there's a high ATP level, maybe genes required in respiration won't be expressed as much, right? You don't need as much ATP in that moment, so you might not need enzymes to make more ATP. So depending on the level of certain nutrients and the cell's health overall, and also chemical signals from the outside, the cell will decide, quote unquote, which genes need to be expressed and which should not be expressed. I'm going to try to use the book's outline. The book says there are 5 primary levels of control. And we're going to use this outline to discuss gene expression, but there's a lot of overlap between these different levels of control. The first couple of levels of control occur in the nucleus. And we're going to start our conversation with chromatin structure. The structure of chromatin will determine whether genes are accessible to RNA polymerase, which will determine which are going to be transcribed and which will not be transcribed. Then there's transcriptional control, which genes are actually transcribed by RNA polymerase, right? If a gene is transcribed, that means it's, could be made into a fully functional protein. That gene will be expressed. But in some cells, we don't want the genes to be expressed. So which genes are transcribed by RNA polymerase in a specific cell is referred to transcriptional control. Post-transcriptional control happens after transcription, and that refers to which mRNAs are spliced properly, right? Introns and exons have to be removed, exons have to be spliced back together, right? That will also have an impact on gene expression. That all happens in the nucleus. Then, after transcription and the mRNA is processed and it leaves the nucleus, in the cytoplasm we have translational control. Which mRNAs are stable enough and protected in the right way so they can be translated by ribosomes in the cytosol. And finally, there's post-translational control. Even after the protein is made, is that protein kept active, or is it rapidly destroyed and inactivated? So, again, this is a summary slide that's very important. We said every cell of your body has the same exact genes, right, more or less. But not every cell expresses the same genes. Based on all these different factors, chemical signals from the outside and inside of the cell could control the structure of chromatin. It can control which genes are transcribed and spliced, right? These signals can control which mRNAs can be translated by ribosomes. And these signals can control which proteins are going to be active and which should be inactivated. So it's a very complicated interplay of signals from outside and inside the cell. But we're going to start inside the nucleus. And eukaryotes only gene regulation starts in the nucleus with chromatin structure. And regardless of gene sequences, the structure of chromatin itself can regulate gene expression. And we say that it can do so epigenetically because epigenetics means above genetics. Epi means like above. Epigenetics is an up-and-coming field. It is very, very important and it's very exciting. And epigenetics discusses how just because you have a gene sequence, it doesn't mean you're going to express it. Right? There are a lot of factors that work outside of genetic sequences, right? There are epigenetic factors that lead to all these things. So, if you recall, we said chromatin consists of DNA wrapped around histone proteins. And chromatin can come in one of 2 forms. Euchromatin is loosely wrapped DNA, and genes are actively expressed in euchromatin. So when DNA is loosely wrapped, genes are actively expressed. Heterochromatin is the other version of chromatin, and that's tightly wrapped DNA, and genes will not be expressed in that case. Right? Tightly wrapped DNA in the form of heterochromatin will not be expressed. How does the structure of chromatin impact gene expression? So I want you to take a second to think about it and actually try to make the connection between chromatin structure and how that could impact the expression of those genes, right? How could it impact the expression of the DNA wrapped around histones? So you could pause here and really think about it. And the answer is, the structure of chromatin dictates whether RNA polymerase can actually bind to the promoter of a gene and initiate transcription. Remember, the first step of gene expression is RNA polymerase transcribing the gene and binding that promoter. If the promoter is blocked because it's so tightly wrapped around histones, the gene will not be expressed. So this is a good schematic. Depending on how tightly the DNA is wrapped around histones, the RNA polymerase may or may not transcribe the gene. If this is the gene promoter, and it's not nice and exposed, then the polymerase can easily recognize this and make mRNA. If it's tightly compacted chromatin, that's very hard to read this promoter. Right? In fact, RNA polymerase won't be able to even bind to it. So this hard to read DNA will not be transcribed. Right? When chromatin is tightly packed, it's heterochromatin, the gene promoters are blocked, and the genes cannot be transcribed. When chromatin is loosely packed, as Euchromatin, for example, RNA polymerase is able to bind the promoter, and the gene will be expressed. I want to actually show you this animation that you can access using this link before you hear. So I actually just realized without Flash we can't use this demo. So what this demo was going to show you was a cell that expresses a glow-in-the-dark protein called GFP. And what you could do is you could control how tightly the GFP gene is wrapped around histones. And you would see that the tighter the GFP gene is wrapped around histones, the dimmer a cell would be, right? The less it would glow in the dark. And as you unwrap the DNA, then the gene would be able to be transcribed into the functional GFP protein, which stands for green fluorescent protein, something that comes from jellyfish. So all we spoke about so far is that the structure of chromatin has an impact on gene expression, because that will determine whether or not RNA polymerase can bind to a gene's promoter. But the next question becomes, what determines how tightly DNA is wrapped around those histones? So it turns out that there are two temporary chemical changes that can happen to chromatin, that can be added to chromatin, that can either loosen it or tighten it. So there are two chemicals. A methyl group is simply a carbon with 3 hydrogens, CH3. If a methyl group is added to chromatin, then the DNA is tightly wrapped around histones. So would that be heterochromatin or euchromatin? Right, it would be heterochromatin. Acetylation is a different chemical group. And if chromatin is acetylated, the DNA is wrapped loosely around histones. So that would be euchromatin. And these, again, these are temporary. So one time, 1 hour a gene could be acetylated. The next hour it could be methylated. Right? So in order for heterochromatin to become euchromatin, there has to be some kind of histone modifications. So what do you think would have to happen in order for heterochromatin to become euchromatin? You would need to have acetylation, right? Remember that would space those space the chromatin out, or you would have to demethylate, right? Remove all of the methylation. And to go from euchromatin back to heterochromatin, you would have to remove all the acetyl groups. You'd have to do deacetylation, and then you would methylate all the histones instead, and that would tighten them up. What does the chemical modifications are enzymes. Specifically, there's an enzyme called a HAT, histone acetyl transferase. Its job is just to add acetyl groups to the chromatin to loosen it up. So a HAT, right, an enzyme that acetylates histones, can turn heterochromatin into euchromatin, basically activating gene transcription overall. A HDAC is a different type of enzyme that would silence genes by removing all the acetyl groups from euchromatin, thus tightening it. into heterochromatin. Right? In this case, the RNA polymerase would not be able to bind, genes would not be expressed. Here, RNA polymerase could bind and genes will be expressed. You don't have to know the exact names of these enzymes, it's just a little extra information. The LALBA gene is responsible for lactation in females after pregnancy. Excuse me. Do you think the LALBA gene is likely euchromatin or heterochromatin for most of a cell's lifetime? So the answer is heterochromatin for most of a cell's lifetime. Would that mean it's methylated or acetylated? Right? That would be methylated. Most of the time, the LALBA gene in every single cell will be methylated. Only in response to certain hormones will the LALBA gene be acetylated and expressed in females. And more so than just acetylation and methylation, chromatin can be completely remodeled. So there are some enzymes that can slide a whole nucleosome over to expose a gene. Some can just eliminate a nucleosome altogether or change the structure. So there are these huge complexes called chromatin remodeling complexes that can interact with chromatin and kind of move the genes around to make certain promoters accessible to RNA polymerase, so the DNA can be transcribed. And up until this point, the methylation and acetylation was done to the histone proteins itself, but the nucleotides of DNA can also be methylated. Specifically, it's the cytosines that are methylated, and certain genes are shut off in sperm and egg. Or I should say, when sperm and egg come together in the early zygote, certain genes are kind of silenced from the dad, and some are silenced from the mom. And it's completely random, as far as we know. This is called genomic imprinting. Right? Certain genes are methylated, certain genes are demethylated. And only one of each gene is turned on in early development. And again, it's enzymes that do this, methylate or demethylate DNA. So I want to introduce this interesting figure over here. And this shows a little paradox in genetic influence. So identical twins contain the exact same DNA. They're clones of each other. And what this shows is the concordance, meaning the percentage of twins that share a trait, whether they're identical, that's shown in white, or fraternal, as shown in gray. So take a second to pause here and tell me what's interesting about this figure, because there's really a lot to talk about. But to save time, I'd rather you pause here and think about it yourself. So nothing has 100% concordance. Even though identical twins have exactly the same genes, none of these traits shown here show 100% concordance. This means just because you have a gene, it doesn't mean that you're going to express it. The higher the concordance between sets of twins, we could say the greater genetic influence. But let's take something like rheumatoid arthritis. All identical twins both have the same genes, right? They all have the same genes in every cell. Yet, only about 10% of twin pairs both have rheumatoid arthritis, right? So just because you have a gene for something doesn't mean you're going to express it, right? Something like diabetes is not very genetic. If only 25% of the time, if you both have the same genes, you're going to both have diabetes. Right, there must be environmental influences. And that's what epigenetics talks about. Epigenetics talks about just because you have a gene doesn't mean it gets expressed. There are so many factors in the environment that influence the epigenome. Whether it's exercise and your microbiome and social interactions and your mood and your diet and the seasonal, all these different things can modulate your epigenome, which can turn different genes on and off. And this is a good thing, right? It adds another level of complexity and regulation to our genome. To some extent, the food we eat modifies our epigenome. Different foods are abundant in either methyl groups or acetyl groups, and having a lot of a certain food type can actually turn genes on or off. So here's a brief overview of the epigenome I'd like you to watch.
DNA contains the instructions for building all the parts of the body. But DNA is only half the story. The DNA in our bodies is wrapped around proteins called histones. Both the DNA and histones are covered with chemical tags. This second layer of structure is called the epigenome. The epigenome shapes the physical structure of the genome. It tightly wraps inactive genes, making them unreadable. It relaxes active genes, making them easily accessible. Different sets of genes are active in different cell types. The DNA code remains fixed for life, but the epigenome is flexible. Epigenetic tags react to signals from the outside world, such as diet and stress. The epigenome adjusts specific genes in our genomic landscape in response to our rapidly changing environment.
So I hope that little animation was helpful, and I'd really, I'm going to require this video to be watched. It's very helpful, very interesting. This has Neil deGrasse Tyson talk about the latest information in epigenetics. And it's only 5 minutes, 5-6 minutes, but I'd like you to watch this news clipping, because it really explains some cool aspects of epigenetics. So which of the following would increase transcription? Pause here. There's two answers to this question. So B or C would increase transcription. Which would decrease transcription? That leaves only two choices. Right, DNA methylation or deacetylation would both decrease transcription. So the next level of control is called transcriptional control, and this is the most critical of all the levels of genetic control we spoke about. So the transcriptional control talks about RNA polymerase, and how does RNA polymerase know which genes to transcribe? We said the genes have to be accessible to RNA polymerase. That's one thing, but still a lot of genes might be accessible. RNA polymerase doesn't just hop on genes and just start transcribing, right? They don't just randomly transcribe genes. Transcription is tightly controlled by proteins called transcription factors. Transcription factors are going to be very important in Chapter 13. It's the transcription factors that can tell RNA polymerase to come to a specific promoter. So transcription factors could say, hey, RNA polymerase, come over here to this gene promoter and start transcribing it now because we need this gene product. And several transcription factors work together to either attract RNA polymerase to a certain promoter or to push it away to repress gene expression. So transcription factors are found in every single cell. Different cell types have different types of transcription factors. So overall, transcription factors can activate or inhibit gene expression. So this contributes to the diversity of cell types and cell functions. Transcription factors first bind to the promoter, which is the beginning of every gene. And then once transcription factors bind, RNA polymerase can then start transcription at the promoter.
By controlling which genes are active, a cell can take on special characteristics. Muscle cells and neurons have the same DNA but perform different functions because they use different sets of genes. A key challenge in genetics research is to understand how transcription factors find the correct DNA sequences to control genes. In the nucleus, DNA is condensed into a DNA-protein complex called chromatin. In places where genes are being expressed, there are often zones of naked DNA. We will now explore ways that transcription factors interact with these zones. Even among scientists, a common misperception is that transcription factors fall from the sky onto the DNA and test one spot at a time. Such a process is too inefficient. Instead, the transcription factor interacts non-specifically with the DNA backbone. This allows it to efficiently search the DNA for its binding site, a DNA sequence it recognizes. The transcription factor slides along until it encounters its binding site. Then it changes conformation to interact specifically with nucleotides on the interior of the helix. Now the transcription factor can stay and interact with other proteins for transcription. Or it can become dislodged by many factors, which could include movement of the DNA or interaction with other proteins. Studies have suggested that transcription factors sometimes move from one part of the DNA to another. Short hops might allow them to bypass other proteins or to hop over compacted sections of DNA. When the transcription factor lands on another segment of DNA, short hops become big jumps. Although the other strand is nearby in three-dimensional space, it may be much farther away on the chromosome, or on another chromosome entirely. There are 1,500 different types of transcription factors in human cells. They interact to create a complex language of gene expression.
So that was an inside look on transcription factors. And mutations in transcription factors can have really odd consequences, like extra digits on the toes or extra wings. Transcription factors are really important in controlling the body plan of organisms and telling which cells, like this cell, you should become hair, you should become muscle, you should become blood. Right? Transcription factors basically turn on different genes and make up each cell's identity, because every cell has the same exact genes. But the transcription factors turn those different genes on or off. That's the key point. So now we're going to talk about transcriptional activators, which is just one type of transcription factor. Remember, transcription factors are proteins. So a transcriptional activator is a type of protein that will bind to a DNA sequence that's far away from the gene, called an enhancer. So an enhancer is a sequence of DNA, usually on the same chromosome as the gene it controls. So a transcription factor has to bind to an enhancer first before it can actually work and activate gene expression. What the enhancer is going to do overall with the activator is increase transcription by RNA polymerase. So an activator has to first bind to the enhancer, which is a sequence of DNA, and then what's going to happen is together the activator transcription factor and the enhancer will loop over to the promoter and increase transcription by RNA polymerase. And again, the enhancers are located far away from the gene promoter, so the DNA is going to have to bend and make it's called a hairpin loop, in order to increase transcription. So let's take a quick look about how these activators work with other transcription factors and the RNA polymerase. So once the activator binds to the enhancer, it is going to loop over closer to where it can interact also with the promoter. This is the promoter in green. So other transcription factors have to be present at the promoter as well. And then there's kind of like a sandwich made. The activator interacts with other transcription factors, and finally the RNA polymerase can start transcribing the gene. Without all of these proteins here, RNA polymerase can't just jump on this promoter and start transcription. We needed the activator to bind the enhancer, right? We needed all these transcription factors to also bind the promoter, and then everything could work together to activate transcription. And just like there are activators that bind enhancer sequences of DNA, repressor proteins bind silencer DNA sequences and can prevent RNA polymerase from binding to the promoter. So it's a combination, right? You can have two sites for activators and a site for a repressor, but the gene will only be made if two types of activating transcription factors are present and the repressor is absent. That's the only way transcription will occur. Any other way, it will not occur, or it will be minimized expression. So altogether, transcription factors work in concert in a very complicated process. So this is an animation included in your book.
Bind to a eukaryotic promoter and initiate transcription. A set of proteins called transcription factors must first assemble on the promoter. The assembly process begins upstream from the transcription start site, where proteins called basal factors bind to a short TATA sequence in the promoter. Other basal factor proteins then bind, eventually forming a full transcription factor complex able to capture the RNA polymerase. Basal factors are essential for transcription, but cannot by themselves increase or decrease its rate. A second set of transcription factors, called coactivators, link the basal factors with a third set of transcription factor proteins called activators. Activators are regulatory proteins that bind to sequences on DNA called enhancers. Enhancers are located at sites that are distant from the promoter. The interaction of activator proteins with transcription factor subunits increases the rate of transcription. Many enhancers, scattered around the chromosome, can bind different activators, which provide a variety of responses to various signals. When a second kind of regulatory protein called a repressor binds to a silencer sequence located adjacent to or overlapping an enhancer sequence, the corresponding activator is no longer able to bind to the DNA.
So that was a really inside look on transcription factors and how they influence transcription. So take a look at this image here that kind of talks about how chromatin is remodeled, exposing the promoter when it's relaxed. And then when it's exposed, different transcription factors can bind to it, including activators that bind to enhancers far away. And then altogether, RNA polymerase and the activators and the transcription factors will transcribe the DNA into mRNA. What is an enhancer made out of? So you can pause here. And the answer is B, DNA, right? An enhancer is part of the gene, technically. And what is an activator? An activator is a protein that's made of C amino acids, because all proteins are made of amino acids. An activator is a certain kind of transcription factor, and all transcription factors are proteins. It's important to make sure this is clear. It could get very complicated otherwise. So take a second to do the pop quiz here. So you can pause Does a heart cell have heart genes? Yes. Does it have heart transcription factors? Yes. So a heart cell will have specific transcription factors that will tell RNA polymerase to go to only the heart genes in the genome and turn them on, so we can make lots of heart proteins. The heart cell, will the heart cell have skin genes? Yes, the heart cell has every gene required. But will the heart cell have skin transcription factors? No. Right? There would be no transcription factors for skin in a heart cell, so a heart cell won't make skin proteins. Right? Same idea. It'll have muscle genes, but won't make the same muscle proteins. And again, I can argue with this because the heart has cardiac muscle. So this is a simplification. And of course, all cells have housekeeping proteins. So make sure this is clear about how transcription factors lead to cell differentiation. Right? How cells become different identities based on the transcription factors they have because different genes are expressed. So now we can talk about after transcription, post-transcriptional control. After the mRNA is spliced, it can leave the nucleus. Splicing refers to the removal of introns, if you remember. So the faster that introns can be removed, the more that gene can be expressed. Splicing can also be done in different ways. So there's not only one way to process an mRNA, and this can add a lot of flexibility to post-transcriptional control. Certain transcripts might include all the exons, while other transcripts might only have some of the exons. So you could actually have different protein products be made from a single transcript of mRNA. So a single transcript may have alternate ways of being spliced, and this is called alternative splicing. So one transcript of mRNA could encode for a hormone called calcitonin. But the hypothalamus in the brain and the thyroid by your throat do very different things, right? They both make that mRNA, but they process it differently. They both make the calcitonin mRNA, but it gets spliced differently, whether it's in the brain or in the thyroid. So this shows you if a pre-mRNA has exons A, B, C, D, and E. Most likely it'll be an mRNA that's spliced to have only the exons A, B, C, D, E, and you're going to get a certain protein product. But it's also possible that an exon might be spliced out as well on purpose. And this mRNA would then lead to a different protein product, but it's still derived from the same initial mRNA transcript. This is called alternative splicing. So again, this is a nice way to get novel ways to express different proteins, more flexibility. Not all RNA encodes for amino acids. I wanted to talk really quickly about non-coding RNAs. And non-coding RNAs can interfere with gene expression. So it's kind of counterintuitive because usually more mRNA leads to more gene expression. But there's some small RNAs that are not coding for proteins at all, and they actually interfere with gene expression. They can turn gene expression off. They can inhibit transcription, translation, or both. This idea is called RNAi, or RNA interference. Certain small non-coding RNAs can interfere with gene expression. So I would like you to watch this brief TED Talk. And I'm going to include the link just for time. I'm not going to show the whole thing here, but this is a really good overview to RNAi. So please look at the link on the next slide and watch this TED Talk. So here's that link to watch right now. So RNA interference deals with two different types of small RNAs. We have microRNAs, abbreviated miRNAs, and we have small interfering RNAs, which are siRNAs. I don't need you to know the details of each of these, but I want you to know they exist. These small RNAs both regulate gene expression by inhibiting it. It usually shuts it off. They interfere with gene expression. So certain parts of the genome don't encode for proteins. They encode for double-stranded RNAs that get cleaved into small pieces. And these small pieces of RNA can actually influence DNA compaction. So they can influence epigenetics and the chromatin structure. These small pieces of RNAs can also associate directly with on mRNA transcripts. So small pieces of microRNAs, miRNAs, can bind to the mRNA transcripts, inhibiting translation. So it can interfere, microRNAs can interfere with translation. Also, it's possible that these small RNAs can be attached to a protein called RISC, the RNA-induced silencing complex. And together, that small RNA and RISC can chop up on mRNAs into lots of different pieces so it cannot be translated anymore. So these small pieces of RNA regulate gene expression by interfering with transcription, translation, or both. So you can watch this animation here included in the ebook that talks about how a strand of mRNA can be chopped up. And then a small RNA can actually interact with an mRNA and cause it to break apart. Very counterintuitive. You wouldn't think that adding a small piece of RNA can stop transcription and translation. But that's what RNA interference talks about. So what does RNAi do overall? The answer is C. It decreases the rate of gene expression. So now we'll start talking about translational control. We spoke about last time how the 5' cap and the 3' poly A tail protect mRNA strands in the cytoplasm so they can be translated into proteins. Once the cap and tail is removed, the mRNA decays and can no longer be made into protein. So in that sense, the cap and tail have a lot to do with controlling gene expression. The longer the tail, the more expression of that gene. Right? So gene transcripts have different survival rates in different cell types, which means different levels of gene product will be made depending on how the cap and tail is placed. mRNAs can also be destroyed by enzymes when they're no longer needed in the cell. These enzymes are called ribonucleases because they chop up ribonucleic acid, RNA. The last level of control is called post-translational control. Even after the protein is made on the ribosome, the expression of the protein product is still regulated in a way. Because if it's not active, right, if the gene product is not active, it's not really being expressed. So this has to do with how the proteins are being activated and the degradation rate of proteins. Because just like mRNAs can be destroyed when they're no longer needed, so can proteins. Specifically, proteases are enzymes that break down proteins once they're no longer needed. So the proteases regulate how long a protein can be active in a certain cell. And these proteases are located in the lysosomes, which are the garbage disposals of the cell. There are also structures called proteasomes in the cytoplasm that are like tunnels that shred proteins. So some proteins that are damaged or no longer needed are tagged with special molecules. And once it's tagged with special molecules, it's doomed and it is shredded by the proteasome into lots of fragments. So we spoke last time about how proteins have to be activated sometimes. And kinases are enzymes that phosphorylate proteins, meaning they add phosphate groups to proteins, meaning these proteins are switched on usually. Usually a kinase adds a phosphate, which means the protein is switched on. A phosphatase is an enzyme that removes a phosphate group from a protein, usually turning it off. So again, this also adds another level of complexity to gene expression. So please watch this video in the Connect site that talks about all the levels of control in gene expression, from the chromatin structure to activator proteins, to the splicing and removal of introns, passage through the nuclear membrane with a proper cap and tail, has to not be degraded so it can be properly translated by a ribosome, and then of course that protein can be phosphorylated or dephosphorylated. So finally we'll talk about gene mutations in the last part of chapter 13. So a mutation is a change in a cell's DNA sequence, and this is a permanent change in the basis of a gene. So in this case, you could say this is a mutant fly because a certain mutation caused the fly to grow legs instead of antenna. So normally, the genes encode for antenna in this specific location on a fly. That's called the wild type. The normal is called the wild type. With the mutation, with a change in the DNA sequence of the fly, legs can grow out instead of the antenna. So that's an example of a consequence of a mutation. Not all mutations have bad consequences. A lot of mutations are harmless. So there are different levels of consequence that can happen from a mutation. Right? No effect at all. It's a complete inactivation of a protein, you can see. A germline mutation occurs in the sex cells, either sperm or egg, which means that every single cell of the developing embryo will be affected by that mutation. That's the most severe. A somatic mutation occurred in one of the developing cells. So a mutation occurred as the cells were developing in the embryo. Therefore, only certain cells of the fetus will have the mutant DNA sequences. So these mutations can't be passed down to the next generation unless the sperm and egg happen to be mutated as well. Mutations themselves can be either spontaneous or induced. A spontaneous mutation is a mutation caused by a replication error by DNA polymerase. So when DNA polymerase makes a mistake, it's just spontaneous. It just happened to happen. So that could happen, of course, if there's a mistake that doesn't get corrected. In the end, only about one mistake occurs for every 1 billion nucleotide pairs replicated. So that's pretty amazing. 1 out of every 10 billion nucleotides is mutated. And most of the time it's not harmful at all. And if it is, hopefully the cell will undergo apoptosis and kill itself. Okay, so it's only when repair doesn't get corrected, when the mistake doesn't get repaired. Induced mutations are caused by a chemical called a mutagen. A mutagen is anything that can mutate DNA. Many mutagens are also carcinogenic, meaning they can lead to uncontrolled cell growth. And there are several environmental mutagens like UV rays and tobacco smoke that we know influence the DNA sequence of genes. So the Ames test is what's used by the FDA to see if a new chemical is mutagenic, if it could cause a mutation in DNA. So suppose you're a candy maker and you want to invent a new flavoring, but you don't want to put it in your candy if it can cause any mutations in DNA, right? If it could possibly cause cancer in that case. So you want to test it on bacteria first. So what we do is we have 2 vials of bacteria, and these bacteria normally require the amino acid histidine in order to survive. Without histidine, they're going to die. So we plate these bacteria on plates without histidine. Excuse me. And normally, the bacteria would just not be able to survive without histidine. But if this chemical is a mutagen, it'll be able to mutate the genome of the bacteria and allow them to survive and grow even without histidine. So if you see bacterial growth, even without histidine on the plate, you know that something in your chemical, in your flavoring, must have mutated the DNA in the bacteria to allow them to grow even without the presence of histidine. Normally, right, bacteria would never be able to grow without histidine. So that's the Ames test, which is used to see if a chemical is mutagenic. So there are different types of mutations, and a point mutation is when one base is changed. One nucleotide of DNA is switched, deleted, or added. A substitution is when one nucleotide is switched to another. So suppose you have a code of template DNA that says 3' ATG 5'. That would normally encode for a mRNA codon that's UAC, which encodes for tyrosine amino acid. But suppose that DNA gets changed into ATA. I want you to look up on a codon table in your book and tell me what would the consequence be if I change the DNA template to ATA instead of ATG. What would the mRNA be? So I want you to do that for each of these possible point mutations. What would be the consequence on the protein if each of these single nucleotides were switched to a different one? And again, this can happen because of a chemical, or this can happen spontaneously because DNA polymerase made a mistake and put in the wrong nucleotide. So pause here and tell me what would happen in each of these three consequences. So there are two types of consequences you can see. A missense mutation is when you have a complete switch of amino acid. One amino acid gets switched to another. And in this case, that didn't happen because ATA still encodes for a codon, UAU, that specifies for the amino acid tyrosine. So even though there's a mutation, it's still the normal tyrosine. So this is a case of a mutation that's not harmful at all. However, it's possible to have a nonsense mutation where an amino acid is switched to a stop codon. And that's what you can see here. If you switch the G to a C, that would encode for a stop codon, and that could cut the protein short. That's called a nonsense mutation. If I switch the A over here to a G, that would encode for a completely different amino acid, histidine, not tyrosine, and that's called a missense mutation, right? The amino acid was switched to another amino acid. So a point mutation could be completely harmless. It could code for a stop codon, meaning that's a nonsense mutation, or it can code for a different amino acid altogether, which is the case of a missense mutation. So frameshift mutations are when one or two nucleotides are either inserted or deleted from DNA. And what that does is it shifts the entire reading frame of codons. So this always renders the protein non-functional. Frameshift mutations are the worst. Suppose you have a sentence, the cat ate the rat. Right? They're all words of 3 letters, just like codons. If I just deleted one letter, the C, and divided up the letters into 3s, I would get a completely different sentence. Everything shifted over, so the entire sentence ruined. If I even add a C, same idea. I'm ruining the rest of the sentence after I add in that C. I'm shifting over all the 3s. So that's what happens with a frameshift mutation, right? Suppose that you add in an extra nucleotide here. That would cause, if you add an extra nucleotide, you can start reading the 3s differently, which can code for a completely different protein. So adding 1 or 2 nucleotides is a lot worse than adding 3 nucleotides, because you would shift all the codons over with the frameshift mutation. And you can learn more about that in this video included in the Connect ebook. So this is the normal sequence: methionine, phenylalanine, threonine, arginine, isoleucine. You can get a base substitution that causes a different amino acid to be put in. That's called a missense mutation. Or you could have a nucleotide added or deleted, which would shift to the reading frame. These are two frame shift mutations, and that would actually be a lot worse because every subsequent amino acid would be switched compared to just one being switched. So this is an example of if you have one sentence, the one big fly had one red eye, a substitution that's missense would be a different letter, right? So you can't read that amino acid the same way. A nonsense mutation would cut the whole sentence short. An insertion of 3 letters wouldn't be that bad, right? If it's a 3-letter word, you could still read before and after the sentence, but it still might change the sentence a little bit. But if you add 1 or 2 letters or delete 1 or 2 letters, then you actually get into trouble because you shift the entire reading frame and then all the amino acids are different. So pause here and tell me what a point mutation is. And the answer is C. What would likely have the worst outcome for an organism? Pause here. And the answer is a frame shift mutation, C. So I'm going to give you, this is an original sequence of template DNA. What would happen if I mutate 2 of these codons into this sequence? Tell me, using this codon table, what would happen? Would both amino acids change, only one, or neither? So take the time to actually do this problem. So hopefully you had a chance to do this and you found that the original sequence encoded for serine and then arginine. But the mutated sequence encoded for proline and then arginine. So the answer is B. Only one amino acid was changed. There was one missense mutation in this first codon, but nothing happened over here. So we saw that mutations may result in a protein's loss of function. That's like an example of PKU, that phenylketonuria, right? There was an enzyme that wasn't working, so you can't break down phenylalanine, and that person built up phenylalanine in their system and causes intellectual disabilities because that enzyme is non-functional. Maybe one of the letters of the DNA code that encoded for the PKU enzyme was mutated, right? Suppose there was a stop codon put in the middle of the gene sequence, so that enzyme was never made, and phenylalanine would never be digested properly. In another type of disease called androgen insensitivity, cells are unable to respond to testosterone because they don't make the proper receptor. So no matter how much testosterone is present, the receptor for testosterone is mutated and it will not function properly. But mutations are not always harmful, and in fact, genetic variation is very important for evolution. And we spoke about that with sexual reproduction, why diversity is so important, especially in changing environments. And mutations create genetic varieties that we humans can pick from, right? We can pick different mutants in fruits and vegetables that favor us, that have the most fruit, or are the most stable, or the most cost-effective. So genetic variation is very important for diversity and evolution. So that's the end of chapter 13. Please complete the lecture review, and this will be covered on the final exam. Thank you so much, and I will see you for the last chapter, chapter 14.